Thank you very much for coming to this symposium. We're very thankful to the RGC for allowing us and also providing us with this TRS award for us to perform this work. Liver cancer is very prevalent worldwide, and unfortunately, over half of the new cases worldwide occur in China, including Hong Kong. And in Hong Kong, it's the third commonest cancer, and in China and Southeast Asia, it's the second commonest cancer. And it has a large healthcare burden, and indeed, it is a very deadly cancer and diagnosed late. And even in tertiary centers, only about 20% of the patients have the tumors operable. And even after operation, there is a very high recurrence rate. And unfortunately, it's also very resistant to conventional chemotherapy. And even with molecular target drugs, there is only a very modest effect. So this is our theme-based objective and goals, the 2 points. One is to understand the molecular mechanisms, how they can contribute to cancer stemness. That is, empowering the cancer cells to be more powerful and more metastatic and more invasive. And the other one is to understand the interaction between the tumor microenvironment and also the cancer stemness. And all this is to devise novel targets and biomarkers that can be targeted therapeutically. So we use a multidisciplinary approach to address the grand challenges and also our objectives and goals, including genetic approach, signaling pathway approach, resistance mechanism, epigenetics, and also importantly, the tumor microenvironment. And this is our research team members consisting of the team, basic scientists working on liver cancer stemness and cancer, and also bioinformatics and also genomics team, as well as clinicians. We also have surgeons and pathologists from 3 tertiary hospitals. And this is the picture of our research team members in our theme-based symposium. And so this is our publications. We have this number of publications, and also quite a number of these are rated as a highly cited paper, and these are our publications. With our experience in the cancer stem cells and cancer stemness, I have been asked to organize a Keystone Symposium on cancer stem cells together with 2 other US organizers in 2021. In advanced HCC, which are not operable, then it depends on new drugs. A first-line drug nowadays is immunotherapy with immune checkpoint inhibitor, and also alternative first-line as molecular targeted drugs, and also second and third lines include molecular targeted drugs and also immunotherapy, immune checkpoint inhibitors. And so to this end, our theme base would concentrate on 2 aspects that's in relevance to the current drugs, including the immune checkpoint inhibitors and also molecular targeted drugs. We also know that the tumor microenvironment plays a very important role because they can interact with the cancer cells to exert or to contribute to more enhanced cancer stemness. So this is the outline of our representative theme-based findings in this presentation. So in 4 areas, number 1, that we identify novel immune checkpoint axis and also novel genes and signaling pathway, and hypoxia and tumor microenvironment, as well as epigenetic regulations, that all these contribute to enhanced cancer stemness. First, on the novel immune checkpoint axis, so using single-cell analysis, we have identified novel immune checkpoint axis in HCC, namely TIGIT, Nectin-2. With using a single-cell RNA sequencing, we particularly look into the immune checkpoints. Particularly for the co-inhibitory immune checkpoints, I'd like to explain a bit, because if these co-inhibitory immune checkpoints are activated, then the cancer cells would then interact with the T cells and suppress the T cell response. Then the T cells may not be able to kill the cancer cells. So in this single-cell RNA sequencing, we identify that this co-inhibitory immune checkpoint axis is upregulated, namely TIGIT and Nectin-2. Sorry. And with experiment and also in vivo confirmation, we identify this TIGIT Nectin-2. It plays a very important role in XUC. And blocking TIGIT or Nectin-2 axis is a novel and promising therapeutic targets for treating XUC. And collaborating along this similar direction, we have also identified a new immune checkpoint pair, namely TIGIT-Nectin-1. Nectin-1 is also named as PVRL1 immune checkpoint pair. And this is an important immune checkpoint pair in XUC, and Nectin-1 is upregulated in XUC. And we also identify that blocking TIGIT can suppress the cytotoxic T cell response, therefore suppressing the activity of the T cells to kill the cancer cells. And therefore, we propose that inhibitors of TIGIT-Nectin plus the single immunocheckpoint or immunotherapy are potential new XUC treatment. So for novel immune checkpoints, we also look into the novel genes and signaling pathways that can enhance cancer stemness. For the signaling pathway, we identified tuberous sclerosis 1 and 2, TSC 1 and 2 signaling can contribute to liver cancer stemness. With exome sequencing and targeted DNA sequencing, we identify a high mutation rate of these two genes amounting to 16% of XUC. And these are inactivating mutations, meaning that they cannot produce a protein. And therefore, the downstream targets or downstream signaling will be upregulated without this suppression. And importantly, this mutation can be detected in routine XUC patient samples. This is FFPE samples. And you can see here, with the mutation, the TSC2 cannot be detected immunohistochemically. And therefore, TSC1-2 mutations can be used to stratify XUCs for personalized therapy. And on another note is that our team has also identified an in vitro hepatocyte differentiation model. So using RNA sequencing on different stages of human ESC cells in different stages of the progress, all the way up to hepatocytes to XUCs, they have identified the different oncogenes that are highly expressed. And tapping into this new knowledge, we have also identified or tapped into this kind of knowledge, also identified different genes that are highly upregulated in different stages. For example, this YALYL is highly upregulated in the liver progenitor specific gene stage. Another signaling pathway I'd like to share with you is that Afrin A3 or EphA2 axis is a hypoxia-regulated tumor-promoting pathway. Using this integrative omics analysis, we identify EFNA3, that is Afrin A3, which is a hypoxia-sensitive tumor-promoting gene. And with further work, we also identified the downstream kinase receptor, EphA2, that's upregulated and also exert the cancer stemness property in XUC cells. And with close collaboration among our team-based group, we identify another receptor kinase in the EphA family, that's EphB2. And EphB2 can endow XUC cells with cancer stemness function. And Nowadays, there's no commercially available EphB2 inhibitor on sale. And therefore, our ongoing work is to test. Already, we have generate an inhibitor. Now we're in the testing stage, is in vitro setting stage, and characterizing its efficacy. And if successful, it could be we could have a leading role in EphB2 in research in the cancer biology. And how about hypoxia and tumor microenvironment? In HCC, because they often grow very large and fast, and it's often very hypoxic. Therefore, hypoxia is a major environmental factor that interacts with HCC cells. And indeed, we have identified that hypoxia would activate hypoxic inducible factor HIF1. And HIF1 can then induce this ecto enzyme, ENTPD2, which then would then turn the extracellular ATP into AMP. And the AMP can suppress the differentiation of the MDSC into maturation. And resulting in the MDSC can immunosuppress the environment and also lead to cancer growth. So the highlight is that using the anti-CD39, which is the same as ENTPD2, synergistically enhance immunotherapy. For example, using anti-PD-1 and anti-CTLA-4 in suppressing HCC. Another factor in the tumor microenvironment is the cancer-associated fibroblasts. And our team has identified that this FSTL1 is pro-inflammatory mediator, and it is highly secreted by activated fibroblasts, that is cancer-associated fibroblasts in the cancer cells. And these activated fibroblasts interacted with the HCC cells to enhance its cancer stemness. And the message is that targeting this STL1 could provide a potential new strategy to improve HCC treatment. One thing about liquid biopsy, which Professor Lowe has also alluded to, and we like to highlight on exosome. So apart from tumor DNA, circulating tumor cells, we work on exosome. And exosome is a very small extracellular vesicle of the size of about 120 nanometer. And now Dr. Judy Yam has identified in HCC patients' exosome, plasma exosome, that the exosome contains polymeric immunoglobulin receptor, PIGR. And this PIGR is present in the plasma, and also it can be confirmed with electron microscopy. And importantly, this exosomal PIGR is highly present in the patients with late-stage HCC. And with further work, she has also identified that this EV, PIGR, can promote cancer stemness through different pathways and also promote the tumor growth and metastasis. And she used an anti-PIGR antibody and showed that it can inhibit HCC tumor growth. And the last, I'd like to talk about the epigenetic regulation. And I'd like to talk about the M6A modification. I'd like to show a graphic here, is that if, for example, a particular mRNA of a gene is added this M6A, it will lead to destruction or degradation once this is M6A added. And we have shown that this methyltransferase, METO3, can add M6A to its downstream gene mRNA and leading to degradation. And therefore, we established the oncogenic role of METO3 in HCC, and we have also demonstrated a new mechanism of epigenetic gene silencing in HCC. We work in a group, and we are strongly supported by our clinicians, clinical oncologists, and also surgeons. And clinical oncologists have published a Hong Kong liver cancer staging system back in 2014. And this Hong Kong liver cancer staging system is very important because it guides treatment and also enable a more aggressive and feasible treatment that is tailored to Asian populations. And this Hong Kong liver cancer staging system has also gained worldwide recognition and application. And also our surgeons have a leading clinical management role in HCC in this region. And our clinical oncologist, Dr. Yao, is also leading many world-leading clinical drug trials on HCC. So we have a very strong clinical support in referencing our work, say, for example, in basic science work and as well as translation work. So our deliverables of our theme base is that knowledge-wise, we have established this hepatocyte differentiation model. We have also identified immune exhaustion in HCC, also identified novel immune checkpoint pairs or axes in the tumor microenvironment, hypoxia, cancer-associated fibroblasts, and immune cells, as well as new knowledge in the RNA modification. We have also identified molecular targets such as listed here, and also precision medicine, for example, using or tapping into TSC1/2 mutation using rapamycin as treatment for these patients, and also exosome with the PIGR. So in summary, our work kind of, we contribute to the scientific advancement, and also this is in cooperation with the clinical advancement. What we wanted is to have integrated omics analysis in more future, and also identify novel gene targets, biomarkers, in order to enhance potential personalized medicine. So we have a very fruitful work in this theme base. These are the joint publications from this theme base, supported by the theme base award. So we have also a lot of graduate students coming from this theme base project, and also postdoc fellows, a lot of awards, for example, the Croucher Senior Research Fellowship, Innovation Award, etc. We have also, from this theme-based research, we have also generated quite a large number of grants and also some patents. We have, in view of knowledge exchange, we have a yearly open theme-based symposium to share our novel findings to the professional communities. This is a yearly event. And also we have held press and media conferences to announce our significant findings of the theme-based research. So our legacy continues because we have been fortunately awarded the second theme-based award, still working, but also advancing into more applicational use, also on liver cancer. And I have been awarded the best female scientist as number 8 in the country and also about 500 in the world. So thank you very much for your kind attention.
Thank you, Professor Ng, for her presentation. So if you have any questions for Professor Ng, please raise your hand and let us know. So Professor, may I ask, so how would you like to translate the basic findings to more clinical usage in the future?
Yes, because our team has clinical oncologists who play a leading role in, for example, clinical drug trials. And we have been working close together, and we will be working with the biopharmas to work with them to see if we can have new drugs, and also we can test the new drugs. They are on the pipeline, and also share under non-disclosure system to share with our findings with them. Thank you. Hi, Tech.
Hi, Irene. That's really amazing. I can't believe just how much work you've done, how productive and how insightful.
Thank you.
One question I have is as the percentage of the HBV-induced liver cancer will drop over the years, and NASH or diabetes associated or not fatty liver increases. Do you have a plan to kind of look at the differences and how you may adopt to the new challenges?
Yeah, thank you, Zach. This is a very, very insightful question. Yeah. Two aspects, HBV-associated HCC. Still, for example, even in Hong Kong, there are still 700,000 people who are infected with HPV. But on the other hand, even in Hong Kong, of course, particularly in Western countries, the NASH-associated HCC is increasing in incidence very remarkably. I think we should also tailor our work to include NASH-associated HCC, and also, for example, in mouse work and also in patients. In patients in Hong Kong, in my own personal experience, we may not encounter a high percentage of NASH-associated HCC in our patient cohort. Yeah, but I anticipate that this incidence will, or this percentage will be climbing up. Yeah, so we are prepared to also include NASH-associated HCC in our study. Yeah.
Do you have any initial insights? Because you have this grant that you have had 5 years, 5 years, 5 years. 50 years later, you're going to have to deal with this, right?
Deal with HCC?
Deal with the NASH.
I do not need to do it.
No, you have to.
Yeah, I think we need to change.
But what are your insights as to the differences in terms of development as well as treatment?
For example, there is still not much good treatment against NASH, but there's very good treatment against HBV. So the HBV incidence will drop, and because there's not much good or efficient drugs against NASH, the incidence will come up. And we have been looking into NASH-associated HCC, for example, in mouse models, using very conventional or well-established mouse model for NASH, and applying our technology like single-cell RNA sequencing, spatial transcriptomics, etc. So we are prepared to shift or include at the stage, but also in the future to shift towards the NASH-associated HCC.
So following Tech's question, does it mean that all your samples are HBV-associated HCC, or do you have information about them?
Yes, we do have information. And in our patient cohort, at least 80% of our HCCs are HBV associated. If we test the patients with HBV DNA, I think it's 90%. So we still have a very high percentage of HBV contributing to HCC. Yeah.
Right. Since your title is about the cancer stemness, right? So I was expecting to see some kind of a pathway or something, but then you have various sort of snippets of results. Not every marker is 100% associated. So can you say something about that? Is that something to do with the HPV or the cancer cell stemness? Because you have not come back to that.
Yeah, thank you very much. Because in fact, in most of the studies that are presented, I think some are purely HPV associated, which I did not highlight. but many of them are HBV associated. So you may say that the results are more on the HBV associated HCCs. Yeah.
So any further questions for Professor Ng? If not, please join me to thank Professor Ng for her excellent talk.
Thank you.
So now we will have a very short break. So please return by 11:30 a.m. Thank you.